slide, I will go very quickly. So most of the time in many other classes, I actually discuss about those. So I will mostly concentrate where it's required. So here. So mostly we will discuss today cell biology, growth and development, as well as if I get time, I will discuss about the development of tumor. Mostly, how cancer developed and why cancer developed. Nobody knows actually. There are many unknown diseases also, but whatever known, the summary of that I will discuss. So now, uh, regarding cell, I several times I told you that there are two different types of cell. One is called prokaryotic. So prokaryotic means like here you can see this is bacterial cell. This is called prokaryotic. Okay, all of you know, and this is eukaryotic. So now here this is one animal cell and this is plant cell. So most of you also know that eukaryotic, this carry-on term in Latin means nucleus. So the thing is, you don't need to remember, but I'm just telling you that in both cases, like in plant cell and animal cell, they have organized nucleus that contain chromosome or DNA. Here also you can see nucleus and that contain chromosome or nucleus. But in case of bacterial cell, prokaryotic, that means before nucleus. So nucleus was not developed, right? So there they have just one DNA molecule. Some bacteria, some exception, they might have multiple DNA, but mostly they have one circular DNA, which is present in the cytoplasm. They don't have nucleus. Okay, that is the kind of one of the biggest difference. But in the next, and you can just go through this slide. You have studied it during your class 9, class 10, all those during that time also. Here, the differences between prokaryotic and eukaryotic cell, I should keep this slide, but I will not go through it. Only one important aspect I should discuss is prokaryotic cells lack histone proteins and eukaryotic cells have histone proteins. There are many reasons for that, but the primary reason that in eukaryotic cell inside the nucleus we have huge amount of DNA, right? Not very small, huge amount of DNA. And in order to keep that huge amount of DNA in a confined and closed space, you need some positively charged entity, because DNA, all of you know, DNA is negatively charged in their, phosphodiester backbone, phosphate backbone, you have negatively charged. In order to, make it stable, histone proteins, those are positively charged protein. The net charge is positive, and that actually help packing of DNA molecule in a small space. Okay, and in addition to that, they have some role in gene expression also. I'm not going into that details, but this is very important point. But prokaryotes, they don't have histone proteins, right? And now other small differences, you can just go through it. I'm not going through it now. Here again, another eukaryotic cell, you can see their organelle in a better way. So this is nucleus. Inside the nucleus, you have DNA. And nucleus, they have some pore. I have actually shared some video with you regarding transcription, translation. If you carefully see this video, you will also see that some pores are there, because it is required. After transcription, RNA should go coming out from the nucleus to cytoplasm. For translation, many proteins need to go inside the nucleus, because that replication and transcriptions are carried out inside the nucleus in eukaryotic cell. But proteins are being synthesized at the cytoplasm, that is outside nucleus. So transportation are required. So as a result of that nucleus, they have pore. Apart from that, we have several organelles. you can just go through it just once. What are the organelles? Then you will remember everything, whatever you studied before in your school days. And
Excuse me, sir.
yeah,
Sir, prokaryote should also contain histone protein to neutralize the negative charge.
prokaryotic cell. Yeah, tell
Should
me.
also contain histone proteins to neutralize the negative charge.
Yeah, prokaryotic DNA, they also have, those are also negative. But the thing is, see, in case of prokaryotic cell, the DNA is, I would say in a small amount, not kind of huge amount of DNA, in terms of their length. Length means base pair, not huge amount. And also they can be little bit relaxed because you don't, like chromosome, as I told you always, that what's the difference between DNA and chromosome? DNA is a kind of extended form. Okay, so that DNA can be stable still because some positive ion like sodium, potassium, magnesium, they also sometimes stabilize those phosphate group, and as a result of DNA can be stable. But chromosome means like very much condensed DNA. So now this DNA is like super condensed. That is called chromosome. That is not happening in bacteria. Okay. So as a result of that, in case of bacteria without histone, still it should be fine. Is it clear, Rajesh?
Sir, but if we leave bare negative charge, then wouldn't it cause problems if it's like positive molecules are transported inside the cell?
Uh,
Or
Other protein you were saying?
no, sir, positive ions like magnesium, sodium, or potassium wouldn't it interfere with the DNA and cause problem?
Yeah, so actually, normally some amount of positively charged ions should be there actually, okay. But the problem in eukaryotic system is like you need one slide I have today also I have one slide that you will see that one DNA molecule how many times it's wrapped around the histone protein and it will make a super condensed structure. During that time, histone is also required because the net charge of histone is positive. Okay, so that DNA can wrap around. So histones are like this bead. So normally one DNA molecule that wrap histone protein like around 2.5 times. Okay, so that way it is wrapped around the histone and many histone together they hold together. That way they make some super condensed structure. Okay, so Rajesh, if you have more questions, please email me. Okay, not the Gmail ID in IIT Gmail ID. And
Yes, sir.
yeah, so now here you will see some of the function of those organelle like nucleus, endoplasmic reticulum, Golgi. Just go through once, right? You don't need to remember all those things, but I believe that all by this time already you know most of their function. Here, this is important. From here, we will start our discussion mostly today. That development of multicellular organism. So as I told you that like after fertilization, that means sperm plus ovum, and after fertilization, they make this fertilized egg that is also called zygote. And then it will divide, divide. That way, it will make some kind of called blastocytes. That means it's more than 60 cells, but around 16, like around 60 cells. But this is not very much defined because it depends on some organism to organism. Like if we are talking about human, then it is like number of cells might be different from another organism in terms of this division. But this is the early stage of embryo formation. Embryo means from this zygote to certain stage, like multicellular stage, that is called embryo. And this embryo will be, implanted on uterus, and that makes, that will be like finally that will develop into full-grown baby. So here you can see this is in this case we are showing in case of human. So this is 4 weeks old, 10 weeks, 16 weeks, and 20 weeks. So by 20 weeks, you can see most of the developmental part like leg, hand, brain, all other things are already developed. Okay, and most of the time, almost all the time, everything is happening in a right way, right? And that's why we are healthy enough. Sometimes, very in unfortunate case, that might be a problem, but most of the time it is okay. So let's see what kind of cell division and how some control mechanism helps this process. We will very briefly discuss, okay, because in one class this is not possible to discuss, but just we will introduce the topic. So cycle of life depends on two modes of cell division, and you guys know that one division is called mitosis and another one called meiosis. These two are different types of cell division, two different types of cell division, and our life cycle depends on both of these two modes. Okay, don't forget these two terms, mitosis and meiosis. This is a cycle, so we have to start from somewhere. Okay, let us start from here. Okay, so sperm and ovum, they fuse together and finally one zygote formed, as I told during last slide. And I also told before that we have half of the chromosome here. So n number of chromosome, because in our body cells, we have 2n chromosome. That means every chromosome we have 2n number. So here also we have n number of chromosome. After fertilization, zygote form, and that is 2n number of chromosome present here. Now, from zygote to adult, that means from one cell to, I always tell you that we have approximately 10 to the power 13 cells in our body. During this process, we rely on mitosis. This one, mitosis. That means from zygote, you are getting like some multicellular stage, and finally the embryo, and finally we are growing up into adult, right? So all those things depends on mitosis cell division. So as a result of that, in our body parts, like whatever you tell, everywhere we have cells that contain 2n number of chromosome or DNA. Why? Because mitosis is nothing but the equal distribution of genetic material and divides one cell into two. So for example, you have one cell. After mitosis, it will make two cells, and both are 2n in number, in their chromosome number, I would say. So as a result of that, when we are growing up into adult, everywhere we have mitosis cell division, and as a result of that, we increase our number of cells. Only in case of like germ cells, that means for the production of sperm and ovum, meiosis takes place, and this is only specific for some germ cell line, not for other body cell. Not meiosis cannot happen in liver cell, in muscle cell, nowhere. Only in case of germ cell, for the production of sperm and ovum, this meiosis takes place. And this is absolutely required so that in the next generation, again, we can maintain the chromosome number. Because the number of DNA molecule, number of chromosomes should be constant in a species. So for example, in human, we have 23 pairs of chromosome, but that should be maintained. So during meiosis, sperm and ovum, they receive half of the chromosome, and as a result of that, again after fertilization, when they combine, again they get 46 chromosome or 23 pairs of chromosome. That way it is being maintained. And I hope that you already understand the role of mitosis and meiosis here. Meiosis is the process where cells get divided, but their chromosome number also get reduced, half of that. So if in some cell you have 2n number of chromosome, like 46 chromosome, after meiosis, from that particular cell, you will get 4 cells, not 2 cells, 4 cells, and each of them contain half of the chromosome. Okay, that is called meiosis. I will discuss little more detail.
Can you please
Yeah.
explain this part again?
Which one? The meiosis, right?
Yes, sir.
Yeah, I have more slides, Tanmay. So like after 2-3 slides, you will find it again. Okay. So that time, please concentrate here. So now, but I believe this part is clear, right? Tanmay?
Yes, sir.
Okay. So here, in order to answer your question, like a little bit more details about meiosis, I will, I have some more slide because we have very important thing to discuss today. That why meiosis is required and what's the implication of that. Because if I cannot explain you that, but I finish this class, then it will be, I would say no worth. So I will discuss that. I must, explain all those things so that you can understand like what is happening. So now, little bit before the cell division, before coming into meiosis and mitosis, I would like to mention one thing that what is happening during cell division. The accurate distribution of DNA. This is a challenging task considering the complexity of our genome. But this accurate distribution is also important because, as I told you, cells without information meaningless. All cells should contain accurate information, and then some cells they work in a different way. That's why they are muscle cells. Some cells work in a different way. That's why they are nerve cells, skin cells. But they have same accurate information, right? Now, if you see each cell contains chromosomes and chromosome contains gene, so you will understand this. So from which way you go, that is fine. So for example, here you have DNA. This I explained before. The DNA nothing but a double-stranded, helix here. This is polymer of nucleotides. And now if you go a little bit in higher order, this is your like chromosome that contain multiple gene, like in one chromosome or in one DNA, you have multiple genes are present, and here one specific chromosome pair. So this is the important thing. Specific chromosome pair means here, as I told you, maybe this is the chromosome, like we have 23 pairs of chromosome. It can be the pair 1, but this is from, for example, from your, from father, and this is from mother. So that's why they are pair, okay? And all together they make 46 chromosome present in our body. I'm not sure how many drawn here. And then all these chromosomes present in every cell, and finally those cells are present in our body. We have that many cells, and during each round of division, everything's happening in a right way. So to add little bit more complexity on top of that, I just added this slide for this semester. How long is your DNA? So in order to answer your question, even if I ask you this question that how long is your DNA, and if I provide you some of the basic information, I believe you can calculate the length of your DNA present in your body. That length is really, really big. You cannot really imagine if you didn't already search in Google or somewhere. The DNA inside each of your cell is longer than you are. Again, I'm telling this sentence. The DNA inside each of your cells is longer than you are. Now you have around 10^13 cells. So now you understand that total, if you add, then how much, like what will be the length of your DNA? But packs down into a space smaller than you can see. You need a microscope to see nucleus of your cell, right? So that's why the DNA is packed in such a way that you cannot see in open eye. But the DNA is almost 2-meter length. The DNA present in just one cell, it is approximately a little more than 2-meter length. So I will show you the calculation here. This is DNA structure, right? All of you know in your first class, I discussed about it. And the length or the distance between two nucleotide here is 3.4 angstrom. And also you should remember that we have 3 billion base pair per haploid set. Haploid set means in number. Like we have 23 pairs. So just in 23 chromosome, we have 3 billion base pairs. Hence, in a diploid cell, it should be 6 billion, right? So 3 into 2, 6 billion. Now, what will be the length? Length of one base pair equal to 0.34 angstrom. So just multiply this, and then 6 into 10 to the power 9, that many bases are there. And finally, you will get approximately 2 meters of DNA present in each of your cells. And how much DNA is an average human body? And this is a kind of approximate answer. Okay. So as a result of that, this is a challenging task to distribute DNA during cell division. But as I told you repeatedly, that almost all the time these processes are happening very nicely. But whenever some mistakes happen, we have very nice control mechanism so that our body can detect the problem and then either try to rectify that, and if it is beyond rectification, if it is beyond correction, then we kill that cell. We destroy that cell. That is called programmed cell death. The cell death is already programmed. That means something beyond repair happened, then better to kill that cell, right? So I have one slide later, which has been dedicated to apoptosis or programmed cell death. I will discuss little later. Okay, just remind this, just remember this small thing, but I will discuss at appropriate place.
Sir, what is haploid and diploid?
Haploid and diploid means in our body, right? All cells, like whatever cells, liver, brain, muscle, kidney, all cells are diploid. Diploid means cells, nucleus, and inside the nucleus you have all 46 chromosomes. Okay, all 46 chromosomes. Sorry. So that means 23 pairs of chromosome present, 46 chromosome. So this is diploid cell, and that I am referring that 2N. 2N means N means here 23. 23 I have received from my mother and 23 from my father. 23 plus 23, 46. That's why 2N. This is diploid cell. Haploid cell. If this is haploid, then we have only 23 chromosome, not the 46. And this is haploid. And this is normally we denoted as N, and this is 2N. Is it clear?
Yes, sir.
Okay, very good. So in adult, in our case, only like sperm and ovum, those are haploid cell. All other body cells are diploid. Some you don't need to remember, but some organisms are there, very exceptional cases that their body cells are also haploid. Only few examples are there, like some honeybees, right? They have haploid cells, right? But those are little bit complex. You can forget now, and if you are interested, you can read little bit more later. But remember that our body cells are diploid, and only germ cells that makes sperm and egg, only during that time we get haploid cells. Now, coming into the discussion that expansion of zygote. Zygote means the single cell which produce just after the fertilization. Expansion of zygote to adult human depends on mitosis cell division, as I told you. And here, before seeing this image, I would like to tell you that division of one parent cell produce two daughter cells. Okay, and the chromosome number remains same after mitosis. That means from 2N to 2N. Whoever asked this question, so now I believe that it's clear to all of you that 2N to 2N, that means the chromosome number should be same, and that's why it is called equal division or equational division. So after division, they should have the same number of chromosome. And here you can see this is a cycle here, right? So here you can see this is a cycle. So cycle means what kind of cycle? This is called cell cycle. So here there are many steps mentioned here which you have studied during your school days. You mightn't have biology in 10+2, but even at 10+ level you actually studied about some of the cell division stages, right? Prophase, metaphase, anaphase, telophase. But those complexity, like just some terminology, I'm not going into that details. Even if you forget, nothing will happen. But you should have proper idea about cell division. So that I am going to discuss little bit in this whiteboard to give you like overall idea about cell cycle. So this is, I'm just making some kind of cycle. So this cycle is going in this direction. Very carefully listen this. I'm just trying to summarize like one big chapter in biology textbook that is called cell division or cell cycle. So now, in case of cell cycle, that cycle means what? It started from here. It's going in this direction. I mentioned the arrow. Again, it is coming here. So in this cell division, we have two major phase. One is called here M phase. This is also called as divisional phase. That means the actual division, that means from one cell to two cells, that actually happened here in this period, right? If you make this total time, that means from here to coming back again here, if it is 24 hours, this M phase is little less than 1 hour. Although many things happen in M phase, that actual division, but the time takes is very less. So let me finish 3-4 minutes, then I will give you time to ask question. So this is called divisional phase. Now, divisional phase means, for example, you have one cells here, just at the starting point of M phase, the cells, and this is nucleus. When it is going through this M phase, now you have two cells, okay, two cells, and they have 2n number of chromosome. I'm not going into that chromosome yet. I will discuss that. So M phase, and the rest of the portion here, whatever I have drawn, apart from this M phase, that's it. That is called interphase. Interphase. This interphase we can roughly divide into 3 different stages. Okay. You might, remember, you might forget, but after going through this slide, you will get similar information in certain slide also. That's why I am trying to explain here, so that will help you to understand. So this thing actually you can divide roughly into 3 phases. So this is, I told you that M phase, and here this one from here to here. So what is this? This is nothing but time, and the cells are going in this way, and then cells become larger, and finally cell will divide into 2. So the first stage is called G1, this one, G1, or why G? G stand for gap 1. The next one here, this phase is called S. S stands for synthesis. And then the last one of interphase is called G2, nothing but gap 2. So then in cell cycle, if you divide a classical, or I would say ideal cell cycle observed in our body, you can make or divide into 2 different phases, M phase or divisional phase, and the interphase. Interphase is made up of gap 1, S or synthesis, and G2. Okay, G1, S, and G2. this way cell is growing. So for example, if you take this cell, for example, whatever cell produced here just after M phase, then what will happen when it will go through this cell cycle? So this cell will grow little bit in size, and it will make proteins and other things inside the cell, and finally the cell will be here, like at the end of G1, cell will be little bit bigger, and all the things are present inside the cell that should be checked whether the cells is properly grown or not. If everything correct, then this cell will enter into the S phase. Do you know this S or synthesis stands for what synthesis? What kind of synthesis we are talking about? I didn't discuss here, but can you guess?
Protein synthesis and
DNA
DNA
synthesis.
replication.
DNA synthesis,
DNA
yes. DNA
replication.
replication, yeah. Here S means DNA replication. Synthesis means DNA replication, because you have here how much DNA? You have 2N. Like we are talking about mitosis, so 2N, that means 46 chromosome present. Here also you have 46 chromosome, 2N number of chromosome. But in order to divide those cells into two, again, so first you have to duplicate your information. You have to double the amount of DNA present, right? So as a result of this S phase or the synthesis phase, the major function, you are right that during S phase also many proteins are being synthesized. I'm not telling that 10 hours nothing is happening, but the major function in this context is the replication, that 46 chromosomes are now getting replicated, and it will make, like, if this is 2N in case of human, because if you are talking about cat, then the number will be different, not 46, right? So that's why we are mentioning 2N. Then this is any animal or any plant. So now the diploid cell, their chromosome will start getting replicated. So during S phase, chromosome get replicated, and finally at the end of the S phase, you will see that you have this cell which has chromosome number. What will be the chromosome number here? Can you tell me, please?
4N.
4N, right?
4N.
4N, because the synthesis already happened. That means the replication already happened. You have 46 chromosome here because 2N. Now it is 4N. Now this cell will grow.
I have one doubt. So,
Yeah.
at the end of the G1 phase, you said that the cell will grow in size. We know that an organism grows by the division of cells only, but how can a cell itself grow in size? Like, do we see division of cell
Yeah,
organelles?
I understand your point. Yeah. You are right, actually. So you could say that that cell will only grow in size during, I would say, in G2 phase, like after. But both happen actually, Tanmay. So what happens? Growth in size is not always in terms of volume, but in terms of mass also you can consider, because during G1, many of those proteins. Say, for example, I would like to tell you, like, lot of DNA polymerase should be synthesized during that time, because just one DNA polymerase is not enough during this replication process. We have 46 chromosomes, and all those chromosomes need to be replicated, and many DNA polymerases are required for the synthesis of ligand strand. So all those synthesis happen actually, right? So that way, this is, you can say, growth. Growth means in terms of synthesis, all other stuff. So as a result of that, both G1 and G2, you can, like as I mentioned here, that gap 1, gap 2. In some cases, people might say growth, like growth also happened here in both cases, right? So because otherwise, I would say after G2, when cell get divided into two, like from one cell to two cells, then from that one cell divided into two, then those two cells will be smaller, but they need to little bit grow also. So that way it is being maintained. So Tanma, is clear?
Yes, sir. And so also another question. So,
Hmm.
during cell division, like we also have to like divide the cell organelles, right? But otherwise,
Yeah,
how
very good
will the
question. Very good
so
question. Yeah.
in
So
which phase does that happen?
yeah, I will tell that. I'm not yet there, but actually, so to all students actually. So whatever we are discussing today, normally we discuss in two defined classes, but this time, like we actually reduce the number of classes. We are taking more time to discuss. Intentionally, we don't want to pressurize you with lot of information, but I will touch that. So in a previous semester, I have like 1 or 2 slides for that, but I will address your question. If I forget, please remind me again. But I will address within this slide itself. Not this slide, in this whatever I'm explaining now. So here this is 4n. That means after the S phase you have the cell which has double amount of information. Now the cell is going through G2. G2 means gap 2. Here there are many other checkpoint. Checkpoint means say when you are driving your car, if you are driving car, scooter, cycle, we are going through some road, you also see road signal, right? Sometimes stop sign, sometimes yellow sign, sometimes caution. So while cells are moving through this cycle, there are many checkpoints are there. And those checkpoints properly actually check the status of the cell, whether the cell is in right shape. Shape means not size shape, I'm saying, that whether everything perfect or not. If not, then that cell needs to be repaired. If it is beyond repair, then cells need to be killed, right? Like unwanted, like very one cell with lot of misinformation, bad thing, then it will divide into two, and then two things will be wrong. Then 2 to 4, it will multiply. So as a result of that, if something is really wrong, then better discard that cell. That cell should be, killed. We have some procedure. We have some very much, like nice process in order to do that. But the thing is, while it is going to G2, it is checking whether the replication happened properly, whether chromosomes duplicated, its kind of the information is really double, and all other materials are ready in order to divide the cell. Yes, everything perfect. So cell will start dividing just after G2, where in the M phase. And this is the more, I would say, many things happened here. Like what things will be happening? The two things, like like two important thing is, chromosomes are already double. You have to equally distribute chromosome into two cells, right? Equally means it's really equally, not just in terms of number. Imagine that you have like 23 pairs of chromosome. Just say the pair one from father, one from mother, and they are now also getting double because of the replication. Those chromosomes should go into two different cells correctly. It is not like that chromosome like pair 1 will go into one cell and all the the replicated chromosome of pair 2 will go into different cell. Then the information will be mismatched. That cannot happen. Exactly equally the same information should be distributed into two cells. So that is the major thing. And the other thing is, while this correct distribution is happening, the cell also needs to be divided, physically divided. So inside the cell, chromosomes are getting separated into two poles of the cell. If you imagine that this is one cell, chromosomes, they are present inside the nucleus, right? Nuclear membrane, they get dissolved during this time. Now, these chromosomes, there are very complex mechanism. I am not going into that. Those are actually prophase, metaphase, anaphase, telophase. So through this method, those chromosomes are going into two different poles. They are moving like this. When they have been already moved into two poles, now the cell will pinch inside. So they will make the division. Now you have two different cells. So one is called the karyokinesis, that means the division of nucleus, and the other part is called cytokinesis, or division of cytoplasm of cell. You can forget this karyokinesis, cytokinesis, this term. So basically you are distributing your genetic material, which has been already doubled, and also you are dividing into two cells. I will show you that complex slide now, and you can appreciate now whatever last 5 to 10 minutes I discuss about it. Now, in order to answer Tanmay questions that how cell organelles get distributed, yes, that's a good question. So during this cytoplasm division, right? During the division of cytoplasm, some small organelles, right? Say like ribosome, Those ribosomes are very important. They help in protein synthesis, but ribosomes are present in huge number. Like even in bacteria, one cell you have more than 20, 30 thousands of ribosomes. In eukaryotic cell, I don't know the right number, but it should be huge. So those small organelles, they just get distributed randomly. It doesn't matter that whether one cell get 55% and the other cell get 45% of ribosome. Again, it will be synthesize more ribosome and it will make a balance. So like small organelle, they distributed in this way, like mitochondria, ribosomes. But how you will distribute your endoplasmic reticulum? Because endoplasmic reticulum is a sac-like structure that you cannot distribute. That's the problem. So during cell division, this endoplasmic reticulum also get disassembled. That makes small bubble-like structure. They distribute and again they reunite and they form endoplasmic reticulum. This is very complex process, but yes, endoplasmic reticulum, Golgi apparatus, all those things, they actually disassemble and again assemble after the cell division. So Tanmay, did you get your answer?
Yes,
Ravely,
yes.
this is the case, but as you understand, we are
Means
not going
the
in.
cell organelles are not exactly equationally divided, but
No, it's not required. Yeah, exactly. It's not required because after the division, again, because you need the information correctly. Because in order to make this ribosome, so who is responsible to make the ribosome, Tanmay? Or anyone?
the
DNA. Yeah,
DNA.
yeah, protein actually, but those are information present in DNA. That's why, like from the first class, I'm telling you guys that the main challenge or the main function is to equally and correctly distribute the information, and also during replication, correctly make the copy of that information. If the information is there properly, then cell will manage, cell will synthesize whatever is required. So that is the important thing. Now you can see whatever I discuss, this is the M phase. This is not in scale. M phase they are showing very big because to show this, the complexity here, this figure actually. But whatever I drawn there, that was more, in time scale actually. So an M phase interface, and that I already mentioned, you will get another slide. So during M phase, you can see this, chromosome which are in dark brown color, they are getting distributed. We are only showing here just 2 pairs of chromosome. But anyway, this is not, you don't need to go in details, but I hope that you understand whatever I discuss in the whiteboard. Okay, so that will be sufficient for you.
I have forgotten the name of those green strands. Can you just tell me the name?
Which one?
So the green strands, the
Green
green
stand
strands.
where? Here.
Yes, so those which are connecting the
Okay.
chromosomes.
This is called microtubule. Okay. Microtubule.
Okay, sir.
I didn't tell that yet. Yet means I didn't go into the details. So the thing is, when the chromosomes, as I told you, chromosomes are getting, going into two pole of the cell before the actual division, right? Then how it will go into the two different pole of this cell? So that is, microtubule. That is again some polymer of proteins, not proteins, polymer of proteins. They have like different proteins and that make polymer. So those microtubules, they pull chromosome, they attach to the chromosome in one portion and they pull it together in the two pole. So as a result of that, chromosomes are getting separated into two poles. And when it's kind of equally, separated, then from the middle cell get divided and now you have two cells, right? So this is called microtubule, which pull the chromosome.
Okay, sir.
Uh, now, uh, here, uh, this is again, like again, someone of you actually asked this question about little bit more about mitosis and meiosis. I will discuss that. One very important aspect like present here, what you need to understand, whether it is like it will be covered in exam or that doesn't matter, but that is the like major important thing about the meiosis. So in this case,
Excuse me, sir.
yeah.
So those microtubules and the spindle fibers are the same thing.
Yeah, you can say, yeah, microtubule is spindle fiber is some kind of, like generic name, that spindle fiber, right? But microtubules is more, microtubules made up of two kind of protein, like, yeah. So spindle, though they are, I would say, the major, so most of the time in cell biology book, you will find it as spindle fiber. You are right, right? But the thing is, their constituent, they are nothing but polymer of proteins, and they get, like this is very complex process, because how they synthesize, how they are, disassembled, because that way, see, you understand, right? They are actually pulling these two things together in the two end, right? So that way, it is actually very much important. Now, here, that one cell, um, two cells, and then meiosis. This is mitosis. As I told you, that you have one cell, and that is called 2n, and 2n to 4n, and then 4n again you are getting 2n into 2n. This is mitosis. But in case of meiosis, this is complex. That you have, like, initially you have 2n number of chromosome. In case of human, it should be 46. And then after the synthesis phase, after the S phase, now you have 4n number of chromosome, right? 4n number of chromosome. And now this 4n, like, this cell will be divided into two cells. Now they have 2n number of chromosome. But now this time, the chromosome present in this cell will not, like, replicated again, but those cells will be divided. So as a result of that, finally you will get 4 cells that contain n number of chromosome. That means you have 23 chromosome. Okay, 23 chromosomes. So as a result of that, all cells, in case of human, after meiosis, you will have 23 chromosomes. And these haploid cells are called gametes. It can be sperm in case of male individual, or it can be egg in case of female individual. But now I would like to mention in the next slide how, like, during this during this process, during this meiosis process, like, whatever one individual has information from his father and mother in terms of chromosome, that also gets shuffled during this meiosis process. And that actually generates diversity, evolution in human also, in any other organism. The meiosis helps in diversity. This is very big term, big understanding. I will spend the next slide, I will discuss the next slide, like 5 to 10 minutes, and then I will give you break. Before that, I just, give me a little bit time. I need to check that one student, actually, Anubhav, already asked me one question, that spindle fiber and microtubule. Let me check that little bit quickly, and then I will come to this discussion. Because I told that microtubule, but I have little bit of doubt. One thing, like 1 minute. Yeah, so Anubhav, so they are actually same thing, but mostly like in a cell biology book, when they mention this process, they write in spindle fiber. But as you understand, mostly like my more interest and working experience in proteins and DNA. So normally I happily I'm saying microtubule, but like chemically, I would say chemically or how they are made up of, they are exactly same, microtubule and spindle fiber. Okay, so those are same. So now,
Okay, sir.
yeah, I just checked because I little bit I was like confused for a second, so that's why I just checked it. So it's fine. So now, like if I go to the next slide, that is very important slide. So please pay proper attention. This is the, I would say, the most complex part also. That here, unique features of mitosis and meiosis compared in this case. We have on the left hand side meiosis and the right hand side this B, panel B is mitosis. Please don't see all those things together. It will be confusing. You might have seen this in your like school days, but most likely, most of the time you might not get the importance of it. I would like to clarify that. So first see the panel B on the right side. So in case of mitosis, you can see here, I am mentioning here that 2n number of chromosome. Although 2n equal to 46, but we are showing just one pair of chromosome because just that will be easy to explain here. So we are showing just one pair of chromosome. So as a result of that, you can just consider this red chromosome, for example, it is coming from father and this one from mother, just for example. So that means it is one pair of chromosome, right? But we are not showing the other pair, just we are showing one pair. So what happened after the S phase or synthesis phase? This DNA will be replicated. So as a result of that, whatever the DNA molecule or chromosome of father present in this cell that received from father, that is now also duplicated. You can see that two strands here, not DNA strand, the two chromosomes here. And here also you have two chromosomes, right? That means whatever I show you in these steps, it is getting duplicated, or you can say replicated. Now this is 4n. Now this chromosome, this duplicated chromosome, they actually lined up individually in the middle of the cell. And then this spindle, like spindle fiber, microtubule, whatever you say, that attaches to the chromosome. I'm not telling you the term where it attaches, that is called kinetochore. You just forget those terminology. You don't need to remember. That actually, those terminology creates more problem and you guys don't want to understand. So that's not my. So forget about that. So now those spindle fiber or microtubule, they attaches to the chromosome and they pull those chromosomes into two poles. And finally, when those chromosomes are in two different poles, then from the middle, the cells start divides. And finally, you have two cells. If you see in terms of their nucleic acid component, they are exactly same. Again, they are 2n, right? And they are exactly similar like this one. So as a result of this, if you say this is the cell A and this is cell X and Y, just I'm naming that X and Y, they are exactly similar to cell A in terms of their genetic component. So this is mitosis. Now, in case of meiosis, something happened which stimulates evolution also. I'm going slowly. Again, this is paternal homologue, that means from father. Maternal homologue. Homologue means the two chromosomes, they are part of the same pair, like pair 1. And that's why the one of the pair 1 and the other one of the pair. So that's why homologue. You can, ignore that. After DNA replication, up to this, they are exactly same, both in mitosis and meiosis. Now, what happened? This is very interesting. That pairing of duplicated homologues, right? Pairing of duplicated homologues here, those chromosomes which are already duplicated, they are lining in this way. See, this arrangement inside the cell and this arrangement is different. So here they are like lined by, they are staying like in the parallelly. And now what happened? They exchange their material. That means whatever this is paternal or maternal, but we are human. Most of our genetic sequences are same. So as a result of that, when these chromosomes are lining up in this way, they exchange their material. This material means, say, you can see in this image, just this is cartoon diagram, that some portion of this red chromosome, they are now here, and some portion of this gray chromosome are now here in this red chromosome. This step is called crossing over. Crossing over means crossing over of DNA. That some portion of DNA going into this chromosome and from this chromosome coming into that chromosome. We are showing just one fragment coming here, but not one. It is happening multiple times, multiple places when this crossing over is happening. But then what will be its significance? I will come to that a little bit later. So after this crossing over, you can see some kind of mosaic. That means this red chromosome, it has little bit of information from this gray chromosome. And here also little bit of information from this red chromosome. And now these cells are divided into two. And now if you see, after another round of division, you have 4 haploid cells. They have half of the chromosome if you compare with the starting material here. And now the beauty here is you can see in these cells, in this haploid cell and in this haploid cell, the genetic material is little bit hybrid now. Hybrid means like they acquired some different material, like that paternal and maternal, whatever present in our cell, they get distributed some kind of, because of some crossing over. That means some exchange happened, and as a result of that exchange actually helps. That helps a lot. So it will, that's why we are not exactly similar to our father or our mother. We have, although we are similar to our father or mother, but still we have little bit difference, because this information they recombine during the spermatogenesis, that means during sperm formation or during egg formation, and that helps to bring little bit of new characters, and that helps in like long time, generation after generation it is happening. So as a result of that, that also stimulates evolution. I am sure that many of you might not get all those points, and you might have question also. Please ask me.
So to what degree the crossing over takes place? I mean,
sorry, say again, Rajesh.
To what degree the crossing over takes place means how much variation can it maximum variation can it lead to?
Yeah. So Rajesh, so this crossing over is not, I would say, almost it's kind of random. So when these chromosomes are lining each other, like, so first, let me explain why chromosomes actually crossover happen. So if this is a chromosome from parent, like paternal, and this is from maternal, they are side by side present. So they are part of a same chromosome pair. So chromosome number 1, for example, this is from father, this is from mother. So since like both are like, we are human. So this chromosome 1 coming from father and coming from mother, almost they have equal information, that the sequence almost same. They have might be 1 or 2 nucleotide differences in some places, few nucleotide, that is fine. But now, as a result of that, because of this sequence identity, some kind of like process happened that is called homologous recombination. I'm not going into that details. That result into crossing over. That means, for example, one part of this DNA coming here and this one here, one part from here to here, here to there. This is constantly happening, but this is not very much, you can say exactly how it is not how, that how much, what's the percentage happening. So it depends actually, and this is random, and that is good. So that means what I'm trying to tell you, this is mixing of information, because finally, what the chromosome I have in my cell, like as I told you, 23 from my father, 23 from my mother, right? But my father and mother, they are completely from two different sources. They have, little bit of mismatch information, little bit of different information. And that is good. Why I'm telling you? Because it is absolutely important. Say, for example, I told you before that most of the information or gene information present, say for example, gene for synthesis of protein A, how many times it's written in our genome? That means in one cell, how many times written? Rajesh, what will be the answer? Say, gene A produce some protein. Okay, that gene A should be present how many times in one cell? Can you answer this question, Rajesh?
Sir, at least one time.
Yeah, at least one time, but it should be mostly it should be two times, because as I told you, if this is chromosome number 1 I obtained from my father, this is chromosome number 1 obtained from my mother, so both are human, right? So as a result of that, the more or less information will be same. So for example, if one gene is present here that make protein A, the same information should be present here also, right? So in diploid cell, we have this information twice. Only information which are present in sex chromosome, that is chromosome X and chromosome Y. I didn't discuss in great details yet. I will discuss little bit later. That is little bit different. But information present in other chromosome apart from the sex chromosome, apart from X and Y, in those chromosome, information present in twice. Why I am telling that? It has some important role. Say that protein which is coming from gene A, that might be very important, right? But as a result of that, say for example, whatever chromosome I have diploid, I have two times this information. Maybe one of the information is wrong, some mutation happened, some problem happened there. But still, 50% of the product should be fine. Again, I am telling you, if in one cell we have two chromosomes of same pair, one gene is wrong, the corresponding counterpart is okay. So as a result of that, 50% product will be same. You will get some functional product. So as a result of that, uh, as a result of that, you will get 50% correct information. And this is required because many times it happens to our body, but still we are fine. But now that is why this is very important thing. You will see that marriages, like marriage, uh, like within the family member is not good because of this also. This will create problem because in the same family they might have the same mistake in the same gene, right? Which is prohibited, right? It is not the correct thing, and that will not help this selection process and evolution. But long distance, like not related by their genome directly. So as a result of that, those two individuals are completely different. So this kind of chances are less. Maybe one gene is wrong in parent, like father, one gene is wrong in mother, but finally, when like in the baby itself, there will be no effect. But if they are related by their genome, like in a closely related family member, then the problem will be both the mutation will be together and then same person will be, wrong thing. That will create more problem. But in normal case, what happened, like if some problem, small problem happen, but still 50% are fine. And also during this recombination process, this crossing over process, we develop little new features, right? As a result of that, we slowly get changed, and that is required, and that is absolutely important also. So this is very important thing to understand, to perceive this thing. So I will suggest you to read little bit when you will get time, whatever the material, some online resources, some textbook, whatever you find. And then if you have more problem, please ask me during the next class. Okay.
So I have one doubt. So you said that we have two copies of the same same gene.
Yes.
for this purpose. So, what if like the wrong gene itself is dominant in nature and the other gene is
I understand your question. Yeah. So that will be problem actually. So I got your question, Tanmay. So I'm not really going into the details of dominant and recessive character, all those things. But I would like to mention here, Tanmay. So there are like many like blood-borne diseases, like some kind of there are many blood-related diseases. So they are actually, if you see. Uh, this kind of scenario happen that some mutation happen in 50, like this is called heterozygote. That means 50% of your genes are wrong, right? But still, since it is not dominant, you are fine, like the particular individual is fine. So during for those thing, for those individual, like in developed country, even here also, like if someone is aware about its genetic component, like some although that is not characterized by some disease, but for some reason they found out that this kind of problem are already there, then like during marriage, normally it is advisable, it is actually required to know whether the like whoever that person marrying the other one, that shouldn't have the same problem. So then the offspring, the next generation should have same person wrong thing. Okay, so this is absolutely important thing. There are this kind of like this kind of counseling, like particularly screening and counseling is required for many blood-borne diseases, right, which are like genetic disorder. So if someone is doing like this, then that there will be no problem in the next generation. But without knowing, sometime accidentally it might happen that both the partner they have the same problem, although they are not showing the phenotype, they are not showing the disease, but in the next generation it will be problem. Okay, so it should be need to be careful for those person. Yeah.
yes,
Rajesh.
and so one
Yeah,
more
Tanmay.
doubt.
Hmm.
Yeah, so the during the crossing over phase, like
Hmm.
we have the some of the DNA material exchange between the maternal and paternal homologs.
Hmm.
So that means the nucleotides are actually exchanged.
Hmm.
So does that involve break like unzipping of the DNA just
Yeah.
like in DNA replication?
Yeah, a very good question. So this question, like when I was student, I was also thinking this way. So that might be a problem. Like you're saying that the continuity of a gene sequence, like it might be problematic. That is your question, right, Tanmay?
Yes, sir.
Yeah. So actually, what happened to all of you, I would like to suggest you, this crossing over is dependent on the sequence itself, right? This pairing and this distribution is actually based on the sequence. So it will be sequence specific. As a result of that, what we are in fear, what Tanmay you are telling, so that will not happen. That this will not make or it will not break the continuity of a particular gene. It will be like exchange in a right position, so that it will be in frame, the codon sequence, everything will be in frame. Otherwise, every time that otherwise the crossing over will be very much deleterious, like crossing over will have more problem. No, it will actually help. So the fundamental mechanism behind the crossing over is homologous recombination. That is more complex thing, I am not going to discuss in the class, but because of that it is not going to happen. Rather, the some sequence, like little bit of information will go from here to there. So some nucleotide sequence might be changed, but still they will code I would say the same product, so that you should be fine. But at the same time, maybe you will see little bit of differences in one or two codons, and that will create little bit of different, maybe one amino acid different, like from alanine to valine, although they are very similar. So that way this will help kind of slow diversity, slow evolution kind of thing. But that will not be kind of drastic things, that will not be a problem like this. If something happened like this, Tanmay, it is good question. It might, I'm not telling you that never it happened. It might happen. But honestly, in those sperm or ovum, if it happens after fertilization, that will not lead into a baby. That is also important. Many times this mistake might happen, but that will not lead to after fertilization. there are a lot of mistake will be there, right? So as a result of that, it will not mature into offspring or into that will not lead into a baby, right? So that is also you should know. So many times we don't know, right? That so this is also true that after fertilization zygote from that doesn't mean always it leads to formation of the offspring. Sometimes it get aborted. So that might be because of some very much kind of problematic genetic conditions there. So as a result of that, it cannot sustain. So that's why it is not developing, right? So that option is also there. I'm just thinking actually. So it is right, but based on your question, I just thought about it. Yeah.
Okay, sir.
Excuse me,
Hmm.
sir. Sir, one small doubt. You said that if one gene is defective, then its counterpart is synthesized. So does that mean that means for a single protein, the protein is synthesized two times, means
Uh,
from a paternal and one from
Yeah,
maternal.
yeah, exactly. So yeah, you are right. So since say for example, one protein, just protein A, for example, in one cell, that protein A coming from two genes, at least two genes, right? Where like from one chromosome, which is coming like paternal chromosome, and the another is maternal chromosome, they have the same set of information, right? That you have that information written twice, right? One in paternal chromosome, one in maternal chromosome, and that leads to protein A. Whatever the number of protein A present in your body, for example, protein A present in one cell, the number is 5,000. For example, 5,000 in number. So approximately you can say that 2,500 from this gene and 2,500 from the another gene, right? Now the question is, if one of this gene is wrong, so that might not give you any protein or some wrong protein, right? So but still you have the 50% still there, which might be useful for you, right? And that is there in our body also. I think many of us, we have that kind of small problem, but we don't see those problems because of this scenario. That is my point. That's why I'm investing time and I'm trying to explain you so that you will have some kind of idea about what is happening, right? If I ask the question in this complexity, right, in the exam, that is not good. Like you might not answer, but you should understand this thing. That is clear.
Yes, sir.
Yeah. Okay, so let me see what is present in the slide. Otherwise, I will give few minutes break. Let me see. So here, just one or two slides. Very quickly, I will go. Then you will take a break. So how 2N genome arises? Multiple time I told you. That's why I'm not we're discussing in more details. So here you can see you have 2N number of chromosome. That means now if you count, you will get 46 chromosome here. This is human, and this is male. This is F female, and this is male. So you are here also you have 46 chromosome. After meiosis, what will happen? In cells, you will have n number of chromosome, like 22 plus X. Again, 22 plus X. Those are egg cell. And after meiosis in male, so you will have sperm 22 plus X or 22 plus Y. Why this XY? Because in case of female, both are like the last pair, both are X chromosome. Those are same chromosome. So it doesn't matter. In both the egg, you have the same genetic constituent. But in case of male, it is different. Sperm might have 22, that those are not sex chromosome, those are called autosome. Those are like like autosome, not the sex chromosome. XY, that define the gender of the individual. So now either you have this 22 plus X in the sperm or 22 plus Y in the sperm. So as a result of that, during fertilization, by chance, if this 22 plus X containing egg and 22 plus X containing sperm, they fuse together, that will make again one female individual. This is 2N. You can see X and X both are there. So this is female. And by chance, if this egg and this sperm, like 22 plus Y, they fuse together, then one X chromosome and one Y chromosome, that will make male. So as a result of that, in case of human, there is no contribution from female side, whether the next generation will be male or female. It is important to know also, right? So there is no contribution from female side, whether this will be male or female in the next generation, because egg cells always contain the same set of information, 22 plus X, 22 plus X. Only it decided by sperm whether it has Y or X chromosome, and on the basis of that, it will be decided whether the next generation, like offspring, will be female or male. And here I'm not going into that. After the break, from here the discussion will be changed, that how from single cell we develop different types of cells and body parts. Okay, so we will discuss something different. So as a result of that, please take a break of few minutes, around 3 minutes, and then I will start discussion.